Well, hello everybody. Welcome back to the new era of Biogeography Symposia. I'm very pleased to present to our next speaker, Dr. Pavel Matos Maravi. He works in the Biology Center of Czech Academy of Science, and he's now going to give us a talk about historical biogeography of neotropical butterflies, understanding how species diversity assemble over time and across regions. Please feel free to ask any question in both English or Spanish, and Portuguese is also okay in the chat. So now, Dr. Pavel, please.
Hello, everybody. I'll just share my screen. Can you see it? Please, can someone?
We can.
All right, perfect. Yes, hello everybody. It's a pleasure to be in this wonderful meeting. I have been already listening to some talks. And yeah, well, my name is Pavel. I'm from Peru, but I'm now working in the Czech Republic as a group leader. And we mostly study butterflies, butterfly evolution and biogeography. And today I will talk about one component of our research, which is historical biogeography. And when we talk about historical biogeography, we refer to species diversity patterns and ask questions like why some regions on Earth or in some continents have more species diversity than others. A classic species diversity pattern is the latitudinal diversity gradient, and in this case, studying the fossil record of living bivalves, we see that actually most lineages of this taxa actually originated in the tropics and then expanded into the extratropics or the temperate regions. And we also see that lineages in the extratropical regions actually live longer in terms of geological time scale compared to the tropical lineages, meaning that tropical lineages might have higher extinction rate, but at the same time, higher speciation rate. So with all this, just to summarize, so there are three main processes, and I have been talking about right now in the last 1 minute, which is origination, extinction, and expansion. And these are the three main processes that we study in historical biogeography. And for people working with organisms with little or no fossil record and relying only on molecular phylogenes, we study questions like speciation, which is related to origination, extinction, of course, and dispersal through time, which is related to expansion. So these three processes are key things to study historical biogeography. We are particularly interested in the Neotropical region, which is the tropical area coming from Mexico all the way south to southern Brazil and northern Argentina. And several hypotheses have been put forward to explain why the Neotropical region is one of the main reservoirs of biodiversity on Earth. And there have been some hypotheses referring to speciation, extinction, and dispersal rates, which can be summarized, or two main hypotheses are called the museum and the cradle of diversity. So people studying molecular phylogenes, for example, found that lineages across the world actually have their ancestral origin in the Neotropical region. But at the same time, the Neotropical region also provide the grounds for really high speciation rates towards the present, for example, in the Pliocene or the Pleistocene. This has been found, for example, in ants, in leaf beetles, and also in angiosperms have been found that the neotropics compared to other tropical regions actually have higher turnover rates, meaning that there is high speciation and high extinction at the same time. So there is a really renewal of lineages through time in the neotropical region. But we disentangle more in detail what's going on inside this amazing biogeographical biome or region. Especially because, as we know, in our continent or in the neotropical region, actually we have a lot of biomes, landscapes, origins, and so on. So it's hard to see that there has been only one process working at the same time for all organisms. We ask questions on historical biogeography, again, on speciation, extinction, and dispersal, particularly on butterflies. And butterflies actually have, the neotropical region is actually one of the main reservoir of butterflies on Earth. And current estimates is that the Neotropical region has 40 to 45% of all butterflies on Earth. How we study that? Of course, butterflies are very beautiful, but they are very delicate, meaning that we don't have so much fossil information from them. So we have to rely to study the past on molecular phylogenies. And we have to infer actual origination, extinction, and expansion through time. So we rely on livelihood-based model comparison, and it's amazing that Isabel already has touched this, so I don't have to explain much detail what's going on with this model. So the DEC model is basically inferring ancestral states at the branch level in the phylogenies, meaning that it's anagenetic changes through time, dispersal or extinction, but also at the cladogenetic level, meaning at the speciation process, which can be divided into vicariants and sympatric subset and so on. But there have been also more like sophisticated approaches in case that we want to estimate jointly dispersal and diversification, because the DEC model only estimates dispersal through time and ancestral ranges. But for example, models like GeoC or classe, GeoC is a kind of subset of the classe model, actually jointly estimates in one single run dispersal, extinction, and speciation. But you can imagine that estimating these parameters is actually very time consuming when you have many areas. So the GeoC model, for example, it's a process that involves only two areas, A and B in this case. So we use those models, and so how we do it? So we go to the field, amazing field work in Neotropical region, sample the study organism and sequence them. So we use molecular data to infer our phylogenies. So with the molecular phylogenies, then we apply these models of evolution and infer ancestral ranges. So in this talk, I will focus on one particular lineage of butterflies called the Brasolini, or the old butterflies, which is like very nice, like also called like old butterflies. They're really big and they can even fly on the task. So it's amazing. It's an amazing group. So for this type of butterflies, we use a total evidence approach, meaning that we combine at the same time in the same analysis, DNA data and morphological characters. With that information, we estimate the species tree with a multi-species coalescent. And why we use that? Because we see that at some places in the phylogeny, actually there has been some gene tree discordances, meaning that some gene trees have different tree topologies than others. For example, if you can see my mouse here, so in the close to the root of the tree. So the multi-species coalescent can deal with such gene tree discordances coming from complete lineage sorting. Once we have the phylogeny, then we need to define the region. So that's also another big topic, like I will not talk about now, but it's how to delimit the areas. I mean, you can delimit areas as fine as you wish. For example, you can define big areas or really tiny regions. But in this case, in this study, we advocate for parametric regionalization. So we have a database of georeference occurrence, and we applied a network approach implemented in the InfoMat bioregions program to actually estimate the best fit regionalization of the neotropics for this particular group of butterflies. So these butterflies are actually mostly occurring in tropical forests, rainforests, and actually they have really high dispersal capabilities. So that's why we were not surprised when the program tell us that is the best fit regionalization for this group is actually very large chunks of regions in the neotropics, encompassing the Mesoamerica, northwestern region of the Andes, the Atlantic forest, Amazonia, and the dry diagonal. Then to estimate dispersal rates through time, we need to kind of simulate the evolution of the butterflies via biogeographical stochastic mappings. So we applied 100 of these stochastic mappings, 100 of this simulation of the historical biogeography based on the parameters that we estimated with the DEC model. And we also applied that not on a single tree, but on a subset of the whole posterior distribution tree. So on a subset of 100 phylogenetic trees. So in that way, we wanted to incorporate also phylogenetic uncertainty into our reconstruction of dispersal through time across regions. So for that, we use again the DEC model implemented in the R package Biogeoverse, but the DEC model can be found also in programs like Lagrange, where it was originally implemented, and RefBase. And we estimated dispersal rate through times as previously done, for example, in Antonelli et al. And also we estimated within area cladogenesis through time. This means that for each split in the phylogeny, we actually count how many times is happening this split within one region only. So we use the formula described in Xing and Ri, and we estimated this cladogenesis through time, which is not the same as speciation through time, because in cladogenesis through time, especially in Biogeoverse, in the DEC model, we cannot take into account extinct species. We cannot really estimate the speciation rates. So that's the main disadvantage of the DEC model. But again, we also corroborated those patterns that we found using the DEC model with another approach implemented in BAM, in the program BAM, which estimates actually speciation through time. So this program doesn't estimate ancestral range evolution, but speciation and extinction through time. And actually we have speciation rates through each branch on the phylogenetic tree. And then we assign to each branch the areas estimated by Biogeoverse. So it's a kind of shortcut. And why we did that and why we didn't apply, for example, the GOC model or the class model in general, because we have 4 areas. With 4 areas, it can be really the computational demanding to apply these class models, for example. So we have to make some shortcut for the moment, and we estimated in 2 different main approaches, the patterns to time. And then we wanted to compare those and see if those are congruent, even though they are coming from different approaches. And yeah, we found dispersal through time, and we found again like a pattern that has been repeatedly found in other organisms, meaning that Amazonia, for example, it's kind of the main source of diversity to the whole neotropical region. But we also see some interesting patterns, for example, that Amazonia and the Atlantic forest actually were connected through dispersal of Brasolini butterflies, actually very old time ago. Very ancient, in ancient times, for example, 20 million years ago. But this connection has been lost within the past 10 million years. And we hypothesize that it's because of the expansion of the dry diagonal. We kind of make a summary based on this race through time. And we see that, for example, in the late Oligocene, around 24, 25 million years ago, actually Amazonia was kind of the source of diversity to the neotropical region, and especially to the Atlantic forest. If you see the arrows, this means the dispersal from one region to another region. Then around 10 million years ago, we see that this connection with Atlantic forest actually kind of like diminished or weakened. We hypothesized it was because of the expansion of the dry diagonal. And very recently, we see a much stronger connection in terms of dispersal between the Mesoamerica and Northwestern Andes with Amazonia, which kind of is congruent with the major uplift of the Panamanian Isthmus. So we have found dispersal from South America into Mesoamerica, actually very, very old, for old times, for example, coming even to the late Oligocene. But actually this was kind of strengthened very recently in the Pliocene and in the Pleistocene. And we hypothesized that actually it was this major event, like the major uplift of the Panamanian Isthmus, that actually kind of drained, and there was like, we hypothesized that there were before like inundations, and this was kind of hampering speciation or diversification, but it was not hampering dispersal into this region. And putting all this together with what has been found in other organisms is kind of like we are already starting to make the puzzle here. So we have that Amazonia is kind of the source of diversity for many lineages, not only butterflies or insects, but also vertebrates and plants as well. But we also have regions on the neotropics that are actually kind of cradles in the way that they have high speciation rates through time, but also kind of attractor, meaning that they receive a lot of lineages from different regions into those. So kind of attractor or species or sink of biodiversity or sink of species diversity. And with that, and I don't want to run out of time, so the take-home messages are that now with model-based macroevolutionary inferences, it's possible to not only estimate ancestral ranges. So in classic studies, we only estimated, okay, the nodes, and we have the ancestral areas on those nodes. But with these new approaches, actually with the stochastic mapping and the new model-based methods, we can estimate also the frequency, the magnitude through time. So this open up more horizons to answer questions, for example, on how a particular region actually obtain its current species diversity. But of course, everything comes with limitations. So in the case of the DEC model, for example, we don't estimate diversification. So the best approach would be to join the infer dispersal, speciation, and extinction using the molecular phylogenetic data. And of course, with organisms with fossil record, it's really much better to infer extinction rates, for example. But if we compare approaches as we did for the Brasolini data and all these approaches, and we see also the same patterns in other organisms, and then we have more kind of grounds to claim some dispersal scenario or diversification scenario. So of course, having no fossil record is a big limitation, but also we can do stuff, no? We can compare approaches and we can compare with other organisms and then have some hypotheses for our study organisms as well. And the neotropical biodiversity was assembled through a varying evolutionary processes. So it was not one single scenario, for example, the places and reproduction. No, it was like a mix of different scenarios across lineages, across species, across regions, and through time, changing through time as well. So with that, yeah, so I'm open for questions, and you say like, yeah, you say like I receive questions in English, in Spanish, and maybe in Portuñol as well.
Great, Pavel. Thank you very much. This was really interesting. We have one question that says, this pattern might be similar in Andean butterflies?
Yes, so in the sample here, in the species attractor, so they were comparing diversity of a group of butterflies from the Andes compared to the non-Andes, in this case, for example, Amazonia. And they were comparing different hypotheses, for example, the museum, the cradle, and so on. But they found that the most likely scenario was that the Amazonia was contributing, or the non-Andean regions were contributing to the diversity found in the Andes, but also there were some instances of diversification in the Andes as well.
Okay, great. I have one question. Have you find some pattern that you can define a specific barrier that is influencing the diversification in your study group?
Yeah, I mean, we found the barriers for dispersal, for example, in the dry diagonal. So around 20 million years ago and before, so there were like actually more dispersal between the Atlantic forest and Amazonia. So barrier in the way of diminishing dispersal, yeah, we found there in the dry diagonal. But there were also other barriers. For example, the PEVA system, which was a big kind of like wetland system in Amazonia, that actually for some butterfly groups were hypothesized that this big mega wetland actually diminished diversification. So there were like maybe more extinction or lower speciation. But in the case of the Brasolini butterflies, actually we found that in this time period, there were more diversification, meaning that, yeah, something has been going on there. So we hypothesized that some of the host plants actually are, for example, palms, acacia, and so on, of these butterflies. So they were actually thriving there. So they were like actually more diverse there. And that's why also maybe for Brasolini butterflies, it was a time of diversification during this PEVA's inundation of the Amazonia, of Western Amazonia.
Great. We have another question that says, researchers trying to succeed to decide biogeographic patterns usually only consider plants and vertebrates. What do you think invertebrates researchers can do to improve this scenario?
Yeah, that's a great question, actually. Yeah, I mean, and that has a simple explanation because, I mean, invertebrates is a huge radiation, like insects only. Like, yeah, it's so diverse that we still don't have complete phylogeny. So for all these methods to work, actually, we need to sample as many species as possible in the molecular phylogeny. So, of course, we can have estimates based on some sampling. For example, we can have crown ages and so on, when the group originated and so on. But to answer this specific question of dispersal and speciation, extinction through time and across regions, we need actually well-sampled phylogenies. And in butterflies, at least, we are already starting to get well-sampled phylogenies, at least in some families like Nymphalidae, for example. So we are actually getting into that. And Nymphalidae alone, I mean, we talk about one family of butterflies, but this family is like 5,500 species. So imagine that time consuming, like so much work that you have to put into that. So I guess that's why we need to work more on sampling species and putting them in the phylogenies.
Yeah, that's true. Well, those were the questions. I wanted to take a moment to thank you very much for your time, for being here and giving us this beautiful talk. Also to remind you all that the next talk will start just in a minute from Dr. Villalobos, and you are all invited to see that presentation too. Also that you can stay in Discord channel to discuss any kind of discussion that may arise from this talk. And if any of the audience is interested in forming part of the next year committee for organizing SBE meeting, you can find that information in our website. Well, thank you again, Dr. Pavel.
Thank you.
We are great in time, so great presentation.
Bye. Thank you.
See you.